Welcome to AP Biology. Today's presentation is going to be on DNA synthesis, also known as replication. So we have an enzyme called DNA polymerase that is going to be involved in copying the two strands of DNA. DNA polymerase can only work in one direction. Because it can only work in one direction, we have to copy our two strands in opposite directions. You see, DNA structure is anti-parallel. So one of the two strands of DNA is laid down in one direction and the other one's laid down in the other direction. And so DNA can only work in what we read from what we call a 3' to 5' direction, and it lays down the new DNA in a 5' to 3' direction. And so because of that, it can only work in one particular direction on the DNA. So on the leading strand of DNA, which happens to be on the one end, up here it would be 3', and on this end 5', the DNA can be made continuously towards the replication fork. But on the other strand, which is 3' over here and 5' over here, we have to copy it in the other direction. That creates a problem because you have to wait for the fork to open before you can start copying. And so the lagging strand gets copied in fragments. So it waits for it to open, then it copies a little bit, then it waits for it to open more, then it copies a little bit more. And so these fragments are called Okazaki fragments. They are named after the Japanese scientists that discovered them. And because we have fragments over here, we have to have a few more enzymes involved in this process. So if you look up the top, the leading strand looks pretty simple. DNA polymerase just zips right along and copies the whole thing. But if you look at the other strand, it's kind of complicated. Okay, so we have primers that have to be attached with enzymes called primase, and they're going to help the DNA polymerase get locked on and start copying. And then you have ligase, which is going to link all of the little fragments together. And you have DNA polymerase 1, which is a proofreader, which is also going to be involved in removing the primer and putting the proper DNA in that region as well. So it's pretty complicated on this side. The lagging strand is a mess. The leading strand looks pretty clean. You have a couple other enzymes involved in this process. Topoisomerase is an enzyme that unwinds the DNA. And helicase is an enzyme that separates the hydrogen bonds so that it pulls the two strands of DNA away from each other so that the other enzymes can have access and do their job. There are mistakes sometimes in this DNA process. You have a situation here where you are basically trying to attach about 1,000 base pairs per second. And if you try to do anything 1,000 times in a second, you're going to have mistakes. I think it's pretty impressive that polymerase the first time through is 99.99% accurate. But if you're copying 3 billion letters and you're 99.99% accurate, that means 1 in 10,000 base pairs is a screw up. And that is a lot of mutations, tens of thousands of mutations. And so we need to be better than that. And so we have enzymes that actually go through. They're called DNA polymerase 1. It's the main proofreading enzyme. Is going to go through and it's going to look for mistakes. How do we recognize mistakes? In the previous video, I told you that when the wrong letters try to bond with each other, you get repulsive forces. Well, that's going to push the DNA apart, like you see in this picture right here. The DNA is being pushed apart and it creates a little bump on the surface of the DNA. Well, the proofreaders go through they recognize that, they snip the bump out, and then DNA polymerase 3 will come through again, fix it, ligase will come through and link it, and then you have the correct DNA sequence where the mistake was. So this happens every time cells replicate. Every time they replicate their DNA, you're going to end up with proofreaders correcting lots of mistakes. But still some are going to get through. Generally, every time a cell divides, about 20 to 30 mistakes get through, but that's better than 300,000 mistakes. And so, yeah, we still have mistakes. It's not a perfect process, but most of those mistakes don't cause too many problems, and we don't have too many issues with the mistakes that pass through. But sometimes those things can lead to cancer and other issues in our body.